So, hello and welcome to today's webinar, Study Design for Non-inferiority and Equivalence, Design Considerations and Sample Size Demonstrated on nQuery. So today's webinar will be exploring some of the design considerations when looking at non-inferiority and equivalence type hypotheses and the type of common study designs that end up showing up when you're doing this type of study, stuff like crossover trials, etc. Before we get started, just a few frequently asked questions. Is this webinar being recorded? Yes, it is, and that recording will be sent to you automatically afterwards later today alongside the slides. So if you miss this or need to leave early, make sure to get those later on and they'll be sent on to you. In terms of questions, feel free to use the questions tab on the right-hand side of your webinar window and let me know if you have any questions about anything covered here. If you have any audio issues or technical issues, do use that as well to let us know and make sure that we can fix those as we're going along. So that covers most of what we're interested in today. So yeah, please let me know any feedback that you have. Okay, so let's get back into today's webinar, Study Design for Non-inferiority and Equivalence. As I said, this webinar will hopefully give you a background on some of the common design considerations when considering these types of hypotheses, which are kind of generally dealing with when you want to evaluate whether a new treatment or drug is equivalent or not worse than some pre-existing treatment, and hopefully give you a background on the sample size calculations available in nQuery for this particular area. Before we go any further, let me introduce myself. My name is Ronan Fitzpatrick. I'm the head of statistics here at nQuery, and I've been the nQuery lead researcher since nQuery Interim 3.0. That's about 5 or 6 years ago at this point. And I've given workshops at places like the FDA, JSM, and similar. And obviously, I do these webinars every month. And obviously, in the current situation with the COVID-19 pandemic, obviously these type of online engagements are even more important than ever. So make sure to take advantage of these whenever you can. So in terms of what we'll be covering today, I'll be covering briefly just the background of non-inferiority and equivalence testing as a whole, just to make sure that everyone here is on the same page. Like, I'm not expecting you to have any pre-existing knowledge of this area, so this isn't the case of expecting you knowing the difference between a mean ratio and a geometric mean ratio or something like that. This is kind of trying to build from the ground up. And then we'll kind of split the main part of the webinar into two parts. One, looking at the non-inferiority testing and some of the design considerations that are more common there, and then equivalence testing and some of the situations there. In the past, there has been some ambiguity between non-inferiority and equivalence testing. Like, equivalence testing had sometimes been used interchangeably with non-inferiority testing, but I think at this point in time that delineation is now much more clear, and so hopefully everyone will kind of know that these terms are kind of where they're appropriate in the current day situation. And of course, we'll have some conclusions and discussion and looking over some of your questions at the end. So as mentioned previously, this webinar is presented by nQuery, your solution for optimizing clinical trials. And it's a complete trial design platform to help make your clinical trials faster, less costly, and more successful, with the primary focus being on sample size determination for your clinical trial. And nQuery has been around for over 20 years now and is used by 90% of organizations with clinical trials approved by the FDA have a license of some type of nQuery. And you can see some of the reviews and companies on this slide here. So let's get straight into the background of this issue. So I suppose the important thing to note about non-inferiority and equivalence testing is that they're really both about if a new treatment is similar to a pre-existing treatment. So, in a standard clinical trial, like a phase 3 confirmatory trial in particular, we're usually looking to evaluate whether our new treatment is superior to either placebo or some pre-existing treatment. So we think that our treatment is better than doing nothing or doing whatever the standard treatment of care is for a particular case. But in this case of non-inferiority and equivalence testing, we're looking at a very different hypothesis, which is instead of evaluating whether our drug is better, we simply want to evaluate that either it is no worse than by a certain degree than the pre-existing standard of care or treatment, or that it is actually equal to some or equivalent to some pre-existing treatment. And of course, this is a very common objective in the development of generic drugs, biosimilar drugs, and medical devices. So for the generics and biosimilars, that's pretty obvious. Like if we create a generic version of an off-patent drug, we just want to show that it's basically the same or is certainly no worse than that pre-existing drug. And for medical devices, once again, it's pretty obvious why. Like if you have a pacemaker, you're probably not looking for it to give different results than the pre-existing pacemakers in the market. Now, of course, I won't go into the day, but there has obviously been some discussion and debate over the level of regulatory scrutiny taken upon these kind of like these equivalency decisions. But today we're going to just focus on the statistical side of if you have a hypothesis of wanting to prove that your treatment or drug or medical device is either not inferior to or equivalent to, like what does that look like in terms of design decisions? So non-inferiority in short is trying to prove that you're not inferior to control. Control here could mostly stand in for standard treatment. And usually if you're looking at a non-inferiority type hypothesis, you're looking at a direct measure of the efficacy of a treatment. So basically something like mortality would be an obvious candidate. Something like blood pressure would probably be relatively a direct measure of higher blood pressure is probably bad in most issues for the case when you're doing a medical intervention, at least. That's the most common situation. And so you have a direction of movement for your treatment that is good. Like X increases is good, Y increases is bad. So you want to have a certain direction. And so if it happens that your drug ends up being superior to the current standard of care, that will still be found to be significant. And there is a lot of interest right now in these kind of multiple hypothesis adaptive type models where you could start off with a superiority hypothesis and drop that down to a non-inferiority if required. But there's maybe, in theory, in future, maybe you could go the other way where it's like, oh, non-inferiority is what we're looking for, but if this is looking promising, maybe we can jump up to that. And of course, you can't do that nilly-willy without affecting the type 1 error. So that can be consideration there. And really, the only information that you need beyond your kind of standard testing scenario for superiority is you need the addition of this non-inferiority margin, which is basically an effect size below which you would find your drug to be inferior. And basically, you don't want, you need to exclude the non-inferiority margin from being possible based on the hypothesis test or confidence interval approach that you'll be using. And so that's a pretty standard idea. It's not too difficult. In reality, for many situations, so for example, if you assume a normally distributed data or groups, then in fact, effectively non-inferiority testing is really just your standard t-test analysis, but with a shifted null hypothesis. It doesn't really affect things. But obviously, if you go into cases where there isn't a variance location independence, that isn't as true, that the null hypothesis has been shifted, can have a significant effect. And then we get into the idea of equivalence. And equivalence is probably more of a controversial approach to doing things. I say controversial, it's obviously very widely accepted and is a hugely important part of bioequivalence studies and similar, but I suppose more controversial because the idea of doing basically the statistical characteristics of equivalence testing are very distinct from that of non-inferiority or superiority testing. And usually in this case, you're interested in some kind of endpoint, which is an indirect measure of efficacy with no, in inverted quotes, good direction. And so, for example, with bioequivalence, which we'll talk about a lot later on, we're mostly looking at the concentration of a drug in your blood. And of course, in theory, you could go maybe increasing the concentration might be good, but we certainly can't take that as an assumption. And so if we're creating a generic medicine, we're much more likely to want the concentration profile to be the same or as similar as possible to the standard treatment or drug of care, rather than wanting it to increase it versus the concentrations seen for that drug. And so for bioequivalence and for similar situations, we're now trying to prove that these two treatments are giving the same or equivalent results. And we therefore need, typically the way this is talked about is that the confidence interval would fall between these lower and upper limits. We'll talk about hypothesis testing equivalent in the relevant section. So I think the main takeaway here is that there's this nice graph here of what these kind of hypotheses look like. And effectively, you can see here that case A is non-inferior in the sense that it is above this non-inferiority margin completely. So the confidence interval is fully above that. Whereas for the second one, you can see that in fact the non-inferiority confidence interval falls somewhat below the NI margin, this dotted line here on the left, and therefore we could not reject the null hypothesis of non-inferiority here. And then we see these three confidence intervals here that fall between our lower and our upper, and in that case, we would find for equivalence. But for this first case, we would not find for equivalence if that was a hypothesis of interest, because of course, you can see that the upper confidence interval limit is above the upper equivalence limit, and so on and so forth. And you can see obviously as the CIs go this direction, they obviously become obviously non-equivalent as the upper CIs are above or complete, or the entire CI is above the upper equivalence limit. So hopefully that gives you an idea of what's going on here. One small note that's common across all these, you can notice that all of these use two-sided confidence intervals. Even the non-inferiority case, which is really more of a one-sided hypothesis. I would note that the confidence limit is at basically twice the alpha level of a test. So if I'm doing a non-inferiority test at 0.05 alpha, or like 5% confidence, 5% significance level, then I would need to use, in this case, a 90% confidence interval if I'm going to use a two-sided interval. If I was using a one-sided interval, obviously they're equivalent, but if I'm using a one-sided hypothesis, which both non-inferiority and equivalence are, then I need to use double the alpha. So 5% significance equals 90% confidence level, basically. That's much more important for the equivalence case to be noted. So let's get into the section about non-inferiority testing. So as mentioned, non-inferiority testing is where the hypothesis test is that the treatment is no worse than standard treatment or control by a specified margin. And obviously we need to select that no worse than by a specified margin. So what is that margin? And this is a huge issue of debate. Like lots of people have been discussing this, and there's various different ways that you can do this. But effectively, what you need to do is select a non-inferiority margin based on a mix of expertise and the prior data that exists. So usually, you want your non-inferiority margin to be a fixed fraction of the original effect. So if we're doing a non-inferiority test, we're obviously assuming that the control agent is a useful agent. It actually does something because we're trying to replicate it. And therefore, the FDA talks about, therefore, we want to have a fixed fraction of that effect as our non-inferiority margin. So like 80% of the original effect, for example. But how much you need that fraction to be will depend on a number of factors, ranging from how much variance there is in the actual effect from the original study or studies or meta-analysis or whatever other evidence is available, from other issues that are outside the effect, which I'll talk about after our example. But I suppose it's just very, it's very common that this would be used for generics or medical devices where we're comparing, but that usually non-inferiority testing involves comparing a treatment to a control. So it's just a two-arm trial, typically with the control being like a reference listed drug or similar, and usually placebo isn't included. But we'll note that that will cause issues, and we'll talk about those in detail when we move on to three-arm trials, which we'll talk about later on in this section. But I think the important thing though is because non-inferiority testing is mostly just a slightly more complicated version of one-sided testing, it's relatively easy to extend to different types of designs like parallel or crossover, but also to different types of data such as proportion, survival, and counts. But I think the important thing to note here is that for the continuous outcome, that extension is pretty trivial if you're making the assumption certainly of like normally distributed data, or log normal data for the geometric mean ratio, but it becomes more complicated for things like the actual just ratio of means, which obviously doesn't have a normal distribution, or for non-normal data where there isn't a variance location independence, which is to say that basically the fact that you're choosing a null hypothesis to not be like the nice trivial case where there's no difference or hazard ratio equal 1, where there are situations like that, does make it more complicated to derive what's happening under the null, as well as the alternative, because now your null isn't a simple case like that. It's like we think the null ratio is 0.8 instead of 1, for example. Just a small note there. But in terms of sample size calculations, for the most cases, you're looking at continuous outcomes, and in most cases, this is a very simple calculation. So this is just a non-inferiority calculation taken from the New England Journal of Medicine, comparing two stents, and where they were looking at late luminal loss in diabetic patients. And obviously, that's what you want to avoid effectively, unambiguously, or effectively unambiguously, certainly in this case. And so what they just wanted to do was to prove that their stent, their proposed stent, was no worse than the standard treatment of care stent used for this treatment. And you can see here on the right-hand side a tabular summary of those results. You'll also notice a little dropout rate here. So the sample size without dropout is 99, but they require 250 after accounting for this dropout rate of 20%. So I'll briefly do the dropout calculation just for convenience sake here as well. So this is nQuery. For anyone who's not familiar with nQuery, the calculations mostly occur in this top left-hand panel. When you select each row, you can see there's like some help card information here, and down below there'll be a verbal summary of the result when we're finished. I'm not going to cover this in detail today. I think like most people here are either familiar with the software or will catch on very quickly from what we're doing here. But just in case everyone is unaware, you have your row definitions here on the far left-hand side, and each column is an individual calculation, and the yellow rows are those that we can calculate assuming all other inputs are used. So in this case, they had a 5% significance level. So if you're using a confidence interval approach, you'd be using a 90% two-sided interval. If you're using a one-sided interval, obviously 5% is fine. And a non-inferiority ratio of -1.6. So if the luminal loss was, if you could not reject the idea that you have -1.6 luminal loss compared to standard treatment, then obviously that's a case where we would need to say that the stent isn't non-inferior to, or it is inferior effectively to the pre-existing stent in this case. And the expected difference, so the hope that they had was that you get basically the exact same results on average between these two types of stents. And this is just a small convenience function saying the difference between these two. That's kind of basically your denominator, if you imagine, on your test statistic. And then they had a standard deviation here of 0.45. Giving a effect size of about 0.356. And this would be kind of considered a medium-ish effect size on the Cohen effect scale, or Cohen standardized effect scale. And then they needed a power of 80%. And that gives a sample size of 99 per group. And you note here there's a small verbal summary, and you can copy and paste that or edit that as needed. And you can see that that's relatively easy to calculate. So for most non-inferiority calculations, there's not really a huge amount of complication involved. Now, like in nQuery, you have a large number of options available under non-inferiority, ranging from cluster randomized trials. We'll talk about three-arm trials in a moment, but obviously for one group, crossover, various different complexities of crossover, Williams designs, and so on. And for for count data, like negative binomials, for proportion type data, and for survival type data. So obviously in those cases, things can maybe get a bit more complicated. So for example, let's say with the log rank test, but the principles are generally the same across these different types of tables. So like this is for a survival case. You can see that most of this is similar, like you have your dropout rates and your expected exponential mean rates in each group, and then you have a non-inferiority hazard ratio versus the actual hazard ratio. And most of these work pretty much the same. So there's there's not a huge amount of difference between these here, but if you are interested in non-inferiority and equivalence testing in the context of non-continuous outcomes like survival, I'd be happy to discuss and talk about those in a future webinar or based on any questions we get today. And as I mentioned here, like they had that slight sample size adjustment for dropout. If we just go to the Windows calculator from the assistance menu and we take 99, we take 99, we multiply that by 2, and then divide that by 0.8. So 99 multiplied by 2 is equal to 198. So that's the total sample size. And then we divide that by 0.8. So remember, with dropout, we take 1 minus the proportion dropout, in this case 20%. So 1 minus 0.2 is 0.8. And so we divide that to get 247.5, which they assume presumably rounded up to 250 in their example to make a nice round number in their case. 248 would have been adequate, but they seem to have rounded up in this case. Okay, so that's kind of like a nice introduction to non-inferiority and kind of gets everyone hopefully on the same page. But I think there's a number of things obviously we could discuss about non-inferiority testing that are quite interesting. And I think like the big debate is about the non-inferiority and margin. So, you know, I've been at a number of conferences where people have talked about dynamic non-inferiority margins based on like the variance that you see in the actual study and whatever like that. I think in general, you know, usually there will be discussion involved with the regulator when setting the non-inferiority margin, and you need to have a scientific case for why, you know, going so far below the other treatment would be an acceptable loss in efficacy. You know, and you know, as well as that idea, as talked by the FDA, primarily about that fixed fraction of the pre-existing or expected like standard treatment effect size. There are many other considerations that obviously would affect how much, you know, inferiority you'll be willing to tolerate effectively versus standard treatment. And obviously, you know, a major way might be the safety profile. Like if you have a much safer version of a pre-existing treatment, then you may make the argument that, okay, maybe this might be slightly worse, but not by a very large amount, and it's very much it's very much safer. And then there could be other secondary endpoints, again, might have improvements on other treatments and stuff like that. That's probably more rare. And then there might be just, you know, just a matter, like, you know, it's just kind of related to safety, but, you know, just like easier administration. So if I'm talking about what the standard treatment is currently done via injection, but you have a new formulation that can be done orally administered, that's a significant saving in terms of the amount of effort required to give the treatment, but in terms of patients' willingness to do it, but also just being able to, you know, have patients self-medicate in that case, and obviously the significant cost and time savings for medical personnel. But I would say in general, from a regulatory point of view, the FDA and certainly the EMA, you know, conservative NI margins are generally encouraged. So if you want to take a very aggressive NI margin in the case of a generic or a medical device, and you want to statistically prove that, like I think, you know, like the best case scenario you're looking for typically is that you want your treatment to do the same thing as the pre-existing treatment, certainly for generic medicine. And so obviously in that case, that implies that a fairly conservative non-inferiority margin would make sense. So I think, you know, other considerations like say that if you have a highly variable drug, which is probably more of an issue with equivalence, as we'll talk about later on. But even in the case of non-inferiority, if you have a high variance type of treatment, that may mean you need a more liberal NI margin to account for that. But, you know, I think, you know, if you are confident in your treatment, you probably don't want to be too liberal with your NI margin if you can avoid it. I think one other issue that we're going to kind of get into when we talk about three-arm trials is that, you know, in the standard two treatment design, where you're just looking at your proposed non-inferior treatment versus standard treatment, you are making a strong assumption that the control effect that you are using or assuming from the original study has been retained. In other words, you're assuming that the control or standard treatment actually works, but not just that it just works, but it works by a specified amount. Since if your non-inferiority margin is based on a fraction of that effect, then it's obviously, if that effect is a different size, or M1 is a different size, then that affects what M2 would have been if you had known that a priori. And that means that, in the FDA guidance, which is included in the references at the end of this slide deck, you may be required to replicate the conditions used to generate that original effect very closely, in terms of the administration profile, in terms of the number of doses given, pretty much all of those other things. If you want to ensure that your standard arm, your control arm, is being administered in a very, very similar way, and obviously that if you're doing that to ensure that the blinded control is kept, you probably need to do the same for the treatment arm or for your generic arm. So that can be a significant burden, because obviously, non-inferiority trials for generic medicines, cost is a major reason why lower cost is a major reason why you're doing this. And obviously you may not have the resources or the time or the personnel to administer the type of programs that exist in the protocol for phase 3 confirmatory trials, where we know a lot of money is expended and a lot of effort is expended to ensure that there is watertight from an ethical, bias and statistical point of view. So that can be an issue that you need to be very careful of, and you may need additional evidence and data for regulatory approval to ensure that your control effect is lining up with what they were expecting a priori. Like if you end up having a control effect that seems different, then obviously that's going to create questions. You may need additional data to kind of ensure that your regulatory approval goes through. And I think both of those are big issues, picking the NI margin and this assay sensitivity effect, that basically, you're still getting the same effect as you were expecting. I do want to briefly mention the existence of the, very closely related to the non-inferiority hypothesis, of these superiority by a margin or super superiority hypotheses. And these are basically a case where, instead of trying to prove that you're no worse than a treatment, you want to prove that you're better than a treatment by a certain margin. So imagine, effectively, if we go back to the very first slide, imagine that this upper limit here is the super superiority or superiority by a margin. Probably super superiority margin rolls a bit easier off the tongue. So imagine this is this. To prove the superiority by a margin hypothesis, you would need the entire confidence interval to be above this upper limit. So I just want to mention that briefly, because most of the statistical issues that exist for non-inferiority testing are basically the same for that hypothesis type. And it is different from, I suppose, the classic, when you hear superiority, you're usually thinking of a inequality hypothesis, which is basically that, this confidence interval does not include this zero value here. But obviously, super superiority is a different idea, and it's becoming, there's some interest in it, and definitely there's been growing interest in it over the last decade or so. But just note that it's a different thing. But that in practical design terms, and certainly in terms of sample size calculations, it's very, very similar to non-inferiority sample size. Okay. So as mentioned, if you have that assay sensitivity problem where you don't know, or you need to assume that the control effect has been retained over placebo, or maybe the old pre-existing treatment, if you're assuming that, that's an assumption you need to put extra effort in terms of replicating previous studies and extra data. Then one solution might be to, rather than just assuming it, let's add a placebo group. And then, because we've added a placebo group, we can directly compare reference versus placebo, prove the hypothesis that reference is superior to placebo, and then compare reference to experimental or experiment. And then if those two are non-inferior or equivalent, if you're doing the equivalence hypothesis, then we have proven that we removed the assay sensitivity problem, because we now know they're both better than placebo, given whatever, even if we've done different conditions, the conditions have changed, but since we're comparing both to placebo, we have that confidence that actually there is a real effect. And now we also have proved that equivalent is equal to reference. And for that reason, this might be considered the gold standard type trial to avoid that assay sensitivity problem. But of course, that's only allowable if it's ethical to do so. Like if this standard treatment is obviously and clearly superior to the placebo, then it would be unethical to subject subjects to the placebo group, unless, like having the condition wasn't considered very serious. So, it may be in the cases of chronic diseases with low risk profiles, or in the case of areas where there's less certainty over the efficacy over the reference. So, for example, in depression studies, that's a clear case where maybe both of those considerations come into mind, and where this type of trial might be preferable. But basically, it's just to note that if you could do this, it removes that assay sensitivity problem, but obviously only if you can ethically do that. If you're comparing to a very successful drug or treatment, obviously you can't do a placebo group in that case. This is really only, this is a better way of doing things if you can do it, but obviously not applicable to all situations. And in this case, instead of testing a single non-inferiority hypothesis, really you're comparing two hypotheses, which is first that you want to prove that either the experiment or the reference arm are superior to placebo. Usually the reference arm is what you're comparing to placebo. And only if that is significant would you then test, is the experiment no worse than the reference, or no worse than a non-inferiority margin, or NIM in this case. But a nice situation here is that some of the work done here is that you can simplify that down really to a ratio of differences test, where you're comparing the effect you're comparing the experiment versus placebo, or E minus P, over reference minus placebo, and you want that effect to be no worse than some ratio of those differences. And therefore you can have like a nice wall type test for the retention of effect using this framework. And you can basically use a very similar framework for means, proportions, survivals, counts, rates, etc. And there's also some nice math you can do to get the optimal allocation for a given alternative. So, obviously this is kind of a two-step hypothesis, but can be simplified down to a single ratio of differences hypothesis if you want to. And then in terms of extending this, there's a number of different papers for a number of different types of data and situations. And obviously this is assuming a parallel type trial. Obviously we have 3 independent groups. Obviously this doesn't really make sense for a crossover type trial. Though obviously, crossover type trials are common for non-inferiority and are obviously quite easy. Quite easy. You could use like a Williams type design for 3 arms or 3 treatments, and that's perfectly fine as well. So this is just an example of a phase 3 trial, like for oral care for osteoporosis. So, in this case, we're looking at a fairly simple case where they've used a normal approximation for a percentage adjusted placebo effect. So this is like effect over placebo. So you'll see here that the mean in placebo is obviously 0 because we're comparing to placebo. And then they're assuming that the experimental arm and mean arm are going to be the same, but they had a pretty generous non-inferiority ratio of 50%. So if the experimental arm would be at least 50% the effect of the reference arm, then they would find this to be non-inferior in terms of using oral calcitonin. But that's the case here because this is considered to be a much easier type of approach, one that maybe they expect patients to be more willing to adhere to versus the standard nasal spray arm, which was kind of the standard treatment in this case in terms of osteoporosis. Postmenopausal, I should say. And so in this case, you can see that they ended up with 133 patients on the experimental and reference arms, but only 84 patients in this placebo group. So obviously in this case, they were pretty confident that, not getting this wouldn't cause any major effects over the course of the study. You can see it's like 1.56 probably isn't a huge effect versus placebo. Obviously osteoporosis is kind of one that it's a chronic disease. It takes time for it to have an effect. So within the time constraints of a clinical trial, it probably wasn't considered to be too harmful to have a placebo group in this case. And obviously it removes the effect of the assay effect. And the fact this is a small effect maybe means that that's a more significant thing to keep an eye out for. So in terms of what that looks like in terms of study design and sample size, in nQuery, it's not a huge leap forward. I think the major difference here is, of course, now that we're not talking about a non-inferiority margin on the scale of the original effects, which is like the difference for the differences. or like the difference between standard and control, like a direct mean difference effect. But instead, the non-inferiority margin is on the scale of this ratio of differences. So the ratio of E minus P versus over, or minus P, or the reciprocal will be fine as well. So in this case, they had a significance level of 0.025. So that's equivalent to using like a 95% two-sided confidence interval, if you want to think of it that way. And then the experimental arm, another arm, were 1.56% each. Obviously, you could argue that using, like a normal approximation for a percentage is not the appropriate way to do it, but this is what the authors chose to do in this case. And as we mentioned, the placebo arm is equal to 0 because both of these effects are compared to placebo. These are basically placebo-adjusted effects. And then, as we mentioned, the non-inferiority is equal to 0.5. And you can see here very clearly it explains that this is about the ratio of the differences. But you could also think of it as like the experimental effect versus placebo, or like 1.56 minus 0 being greater than this margin multiplied by reference time minus the placebo effect. And then in this case, the common variance is 6.25. Note that they had the standard deviation in their calculation, but obviously that's just the variance is just the standard deviation squared. So 2.5 squared is equal to 6.25. And then we get into the allocation ratio. So in this case, they had an allocation ratio. They don't actually provide it directly in the paper, but we can obviously infer it from the kind of numbers that they gave in terms of the actual. We know what the calculations gave them at the end, and we know that basically they had ratios of 0.38 and 0.38 in the experimental and reference arms, so there'll be equal sample size in these two arms, and then 0.24 in the placebo arm. So the placebo arm is like smaller than this other arm here by about 2/3 of it, effectively, of the other effect. They needed a power of 80%, and that gave a total sample size of 352. Now, no, actually, they gave 133 per group for these two arms, but I verified this against the paper and the other, like the original methods paper for the sample size calculation, and it seems like they may have used maybe slightly different values for this or something, or they rounded down instead of up. But this, obviously, in nQuery, we always round up, because that's the safer thing to do to ensure that the power is always above the original target power. But for practical purposes, this is effectively the same. I suppose one small thing to note here is that, I talked about there that there's this optimal allocation, which in this example is actually pretty simple. So we can actually just do it very quickly here. So you can see that when we selected this row, you can see in the suggestions, it gives the optimal allocation for the normal case with common variance. And you can see that it assumes that 50% of people should be assigned to the experimental group, and then something related to the non-inferiority margin should be the other two groups. Now, in this case, it's very trivial because the non-inferiority margin is actually 0.5, and it actually means that we have equal allocation for the reference and experimental groups. So they're 0.25 respectively. And you can see that the total sample size in this case is about 26 lower. But obviously, we now have less people in the reference arm. So if the reference arm is important to you, that may not be appropriate. But in terms of having the lowest sample size for the highest power, this is technically the best thing to do in this situation. And like the help cards for different situations, like for survival and proportions and the other endpoints, the count endpoints, they have different definitions of the optimal allocation, of course. So those are given in the respective help cards there. Okay. So hopefully that's given you a decent background on non-inferiority testing. And of course, non-inferiority testing is usually kind of easier to get your head around because, as I said, it's kind of more or less an extension of one-sided hypotheses. And usually you're looking at effects that are real, effectively. Like in this case, we're looking at, this reduces something that, like, this reduces or increases something that we want to increase or reduce. Objectively, it's just good that we want this to happen. Like we don't want to lose luminal, we don't want to lose luminal, we don't want to lose, like we don't want to have luminal loss. We don't want, to have a worse situation here on the effect on osteoporosis. So, these are cases where it's pretty obvious what's happening. Whereas for equivalence testing, it's not always so clear. Now, you can do equivalence type testing, and certainly more people should consider doing equivalence type testing on actual effects, like where you have a real effect. Obviously, we're just moving into equivalence testing here now. And there's certainly many cases, particularly in the non-clinical trial area, where people have inferred that a non-significant p-value on a superiority hypothesis is equivalent to making an equivalence outcome or suggestion. So, a classic mistake that people have made in research is that they go, okay, I'm going to test my two treatments. I'm going to do my standard two-sample t-test for inequality or superiority. And if I get a non-significant p-value, that's the same thing as saying that I think that these two treatments are the same. And that's not true. Like, it's not true. All that you've proven in that case is that that you can't reject a null hypothesis of superiority. That is not the same thing as accepting that they are the same. And if you want to say these two things are the same, then you need to set up a null hypothesis that's basically the opposite of that, which is where that these are inequivalent treatments. And you can kind of see that in this first little like this image here. This chart here, like we want the alternative hypothesis. If what we're trying to prove is that our two treatments are the same or equivalent, we need the rejection region to be around 0, not the acceptance region. Like you can't just invert a null hypothesis like that and use that to prove things in the hypothesis testing idea. That's just not likely. But of course, you can see here this is very different to what you're expecting, because usually, I suppose, we usually expect the acceptance region of the null hypothesis to be bounded, but in this case, it's the acceptance region is unbounded, and the rejection region of the null hypothesis is bounded here, and that creates all kinds of interesting debates. And certainly going back in time, if you read the original papers, there was a lot of debate about how one would accurately do this type of testing. And of course, it became the big issue when we talked about bioequivalence, because as I say, in the early days, non-inferiority and equivalence were kind of used interchangeably. But when bioequivalence testing was brought forward, apologies if you have any interference in the background, I live near a major thoroughway. Bioequivalence is a situation where obviously we can't just assume that concentrations being higher is better. Obviously, concentration being higher could be more toxic or could be less effective. Like that's why we do dose finding studies at phase 1 and phase 2 for toxicity and efficacy respectively. And because we're looking at an indirect measure in bioequivalence studies, we're looking at the concentration of the drug, or maybe the active part of the drug, in blood concentration. We're not looking anymore at the direct effect of the treatment. We're not looking, does it decrease mortality? Does it decrease blood pressure? Does it increase, the other measures that we want to increase? So if we're going to use this bioequivalence approach, which is obviously very standard for the approval of regulatory in regulatory context for generic medicines, then this is very important. So it can be used for direct measures, but obviously we're going to focus mostly here today on the bioequivalence case, where we're kind of looking at indirect measures. And so, as I mentioned, there was a big debate about how to do this, but I'd say basically one approach that kind of caught on early was the 2 one-sided tests approach. And most of the sample size calculations that you'll do in this area use the 2 one-sided tests framework. And effectively, the TOST framework, TOST from Sherman, it says what it does in the tin. It does what it says in the tin, I should say. Basically, you're going to test 2 one-sided hypotheses. Firstly is, are you less than the lower equivalence limit? And then test, are you greater than the upper equivalence limit? So let's go back to our very first slide to get a better idea of this. So, first let's test, is this confidence interval basically above the lower limit? Or, in hypothesis tests, we're just checking, is the difference, can we reject the idea that this is not below this one-sided hypothesis? But then We then test, or you could do it in the other order, of course, but both have to be both have to be significant in this case. And then we'll test, is it below this other limit? So if it's above the lower limit, but also below the upper limit, then we conclude that it is within the two limits. And you can see there that basically the H1 therefore is that if we can conclude that if we can reject both of these hypotheses, then we can find for the alternative hypothesis that it is between the lower and upper limits here. Like basically delta here just stands in for the difference or effect size. So as I say, these are both tested at a one-sided level of alpha, but it's important to note that the overall type 1 error, the overall significance level, is equal to the one-sided level of each test. So this is a general point, but if you have what we call conjoint hypotheses, which is to say that both null hypotheses have to be rejected to find for your alternative, then the alpha for those two tests will be retained rather than, we don't have to, apply multiplicity adjustment. So if both have to be true, then the alpha used for both is the overall alpha for our test. So, if I'm doing both at 5% significance level, then the overall type 1 error is equal to 5%. But it's relatively easy to show that the two one-sided test approach is equivalent to the confidence interval method approach proposed by Kirkwood. Now, there's technically, back in the day, there was a debate between the Kirkwood approach and the Westlake approach, where the Westlake approach, you calculated your confidence interval, but you had to center it on the null effect, or like on zero, or basically there's no difference. But today, basically, most people use this, where you just use the actual confidence interval approach. Like you calculate your confidence interval like you would always, and then see if it's between your two limits. But one important thing to note, as I mentioned very much earlier, but I'll emphasize it here because it's obviously a bit more confusing for equivalence testing, because obviously we now have these two limits, and you're kind of between two things. You're kind of like, that's a two-sided test, but it's not a two-sided test. It's a one-sided test overall. And therefore, you need to take 2 times your alpha for your one-sided tests to get the equivalent confidence interval. And so the classic case is that if you want a type 1 error of 5%, a significance level of 5%, then you need to use a 90% confidence interval. So that's very important. Now, obviously, I'm presenting these as if these are the only way to do this type of testing. They aren't. There was a lot of debate, and there continues to be some debate about different approaches to establishing equivalence, because this closed rejection region, this idea of these being the same, it's not very intuitive. It creates a lot of issues, and I think the solution here is mostly presented because it's kind of the issue that creates the least problems effectively. For these other approaches, they're not really widely used. This TOST/confidence interval approach is basically the standard in this case. And you just notice here, like in terms of bioequivalence in general, I'm not going to talk about it much in general. Like we have these terms like Cmax and AUC, but just a note, basically what we're looking at here is we're looking at the concentration of drug over time in the bloodstream, and then you're comparing versus your two treatments. And then these AUC is basically just the amount of this area under the curve. It's basically just like the area of this curve here, sometimes measured as the area after the maximum. And Cmax is just the maximum concentration, so that will be here and here. And to display this even more, like here at nQuery, we have a legacy product called Equiv Test. It's not under active development right now, so I won't talk about too much. But this is the kind of an example of what this would look like in real life, where we'd select our maximum concentration here, and it would calculate basically the expected drug concentration profile over time using the parametric modeling approach that's used for this type of hypothesis testing effectively. That's the little green line here. Apologies if you can't see this. This is kind of old software, so it's not as visually appealing. And you can see you get your AUC type measurements here, and so on and so forth. And there's other measurements such as Tmax, like the time it takes to get to Cmax. But I think that these are probably the two major ones. And these are basically if you're doing a sample size calculation, those are basically what you're going to be powering for. So in terms of other equivalence issues, I won't cover this in too much detail because it could be a whole webinar by itself, but just to make sure everyone's on the same page, like there are different definitions of equivalence. Average equivalence is mostly what you're interested in if you're looking to get regulatory approval for generic medicines and so on. But there is individual and population equivalence, and these might be cases where you actually want to administer, in the general population. And then, of course, there are options for different measures of equivalence, like the AUC and Cmax. Crossover trials are probably the standard way of doing bioequivalence studies. And crossover trials are obviously cases where the same person is given multiple treatments, and obviously you have to have like a washout effect or a washout period to ensure there's no carryover. Basically, you want to ensure people are back down to their base, let's say, blood concentration before giving them the next drug. But the 2x2 is probably still the classic version, but replicates such as 2x3 or 2x4 are also quite common. So that's like where you might have different, you might do the different, rather than having AB, you might have ABBA or somewhat similar. And then Williams designs will be required if you're looking at like more than 2 treatments at a time. So replicates are mostly of interest if we're dealing with this last issue here of highly variable and NTID type drugs. And these are cases basically where either there's a very large amount of uncertainty, a high amount of variance in the outcome, or for NTID, we're looking at cases where very small changes in concentrations or profiles cause disproportionate effects on real world safety or outcome. So that's a bit of a narrow index drug. And those, just to note that the HVDs or highly variable drugs and NTIDs have different requirements from a regulatory perspective in terms of what type of designs are preferred and what type of equivalence bounds are used. And the FDA and EMA have different perspectives on this, so it's very confusing. But if you're interested, there are references, and I'm happy to talk about that as well. But for most cases, the standard is that we'll look at for a bioequivalence study, we'll be looking at the geometric mean ratio for either the AUC and the Cmax or one or the other. For sample size calculation, certainly we'll be looking at one of these. And we want that to fall between a lower limit for the geometric mean ratio of 0.8 and an upper limit of 1.25. Obviously, if we're looking at ratios, then we're looking at, if we want balanced bounds, we want them to be the reciprocal of each other or 1 over each other. And I think, just to mention, geometric mean ratio just means it on the log scale effectively. So we're logging our AUC, we're logging our Cmax, and then comparing the ratio of the logs. And that like if you're looking at the ratio of logs, then you're looking at normal distribution effectively, because these are assumed to be log normally distributed. And so when it comes to sample size calculations, it's not hugely different from what we've looked at previously with non-inferiority. Except that what we'll typically be getting is this coefficient of variation, which is obviously just basically how much variation you have versus the mean value. It's the classic definition of that, but it gets a bit more confused because obviously we're looking at the log scale, not the original scale. But nQuery does provide a nice little table to kind of get around that confusion. But let's just replicate the bits that are relatively easy, which is that the significance level is 0.05. So we're doing both one-sided tests at the 5% level, which is equivalent to saying we would want a 90% confidence interval to fall between the lower equivalence limit of 0.8 and the upper confidence interval limit of 1.25 for the mean ratio. And in this case, they wanted an expected ratio of 1. So obviously, the ideal situation is, the ratios are the same. Like, the AUC in both drugs is the same. The Cmax is the same in both drugs. But in these calculations, it's often typical. They might pick something like 0.95 or its reciprocal. So just to note that, we're using 1 here, which is the ideal situation where you'll have the maximum power, but oftentimes you might be asked to kind of consider what would happen if it was 0.95 instead. And then we get into the crossover ANOVA. So just to note, the standard approach for doing this type of trial is to use a crossover trial, to use either a mixed model or an ANOVA type analysis. The ANOVA type analysis is probably the more classic version. And then you have your square root MSE from the ANOVA for the error effect. And you can calculate that. You can see that there's like some notes here about where that's derived from, but you can see there's a direct relationship here between it and the coefficient of variation. But instead of having to calculate it by hand, we can go to the assistance menu and the standard deviation option here, and we can go to from coefficient of variation. If we click OK, then that will open a little side table here, or like a little table here, where we can translate a coefficient of variation into the estimated standard deviation on a log scale, which, once again, maybe a little bit confusing, but that's basically equal to this, not this. Like, this is really the standard deviation of the basically the within subject error. Which we could then talk about in the more standard, say, like t-test equivalent standard deviation of the differences, which is different again. So in this case, the coefficient of variation was 0.383, I believe, giving a estimated standard deviation of 0.37. And so, like, if you're wondering what like a HVD is, I believe that's around 0.5. And this is on the proportion scale. You'll often see this given on the percentage scale, so like 38.3% coefficient of variation. But that in nQuery, we're nearly always looking at the proportion, not the percentage scale. So just note there is a translation. You need to go to proportion in nQuery, not the percentage scale. So we input that there, and you can also see we automatically calculate this standard deviation of the differences, and then we enter our power of 80%. And in this case, you can see that we'll get the same sample size as required by these authors. So you can see here, this is the coefficient of variation and so on. In this case, it gave it on the proportion scale, but remember, this could have been on the percentage scale, like 38.3%. And you can see that they require 25 per group, or 25 per sequence, I should say. And there's 2 sequences. It's a 2x2 trial, and that's equal to 50 subjects. So 25 by 2 is 50 subjects, because obviously we have 2 sequences in a 2x2 crossover trial. And so hopefully this, like, this bioequivalence study compared to Viagra, this seems to have given equivalent results. So, in this case, like, this is what they were trying to prove. And obviously, in this case, if the geometric mean ratio of the AUC and Cmax were within that, then you would get that. Now, technically, if you're doing both, really, you should be doing 2 tests, and you need both of them to be significant. Then the power would technically be lower. But I think for most like sample size calculations, and for bioequivalence studies, usually sample sizes are pretty low. Most people just power on one of them, but in theory, if you had a correlation between the AAC and Cmax on average, or the mean ratio of them versus like control versus treatment, or control versus generic, then the power will be lower on average. But if they're independent, I think you'd still have power of around 65%. So it's not the worst type of situation here. But just a small note there, I suppose, because if you have a conjoint hypothesis that both of these will be significant, then the power will be lower. So as I mentioned, the alpha is not affected, but the power is lower, and that's basically true here as well. Like, the fact that we're doing 2 hypothesis tests means that the power will be lower than if we just needed one of these to be correct. That kind of trivially seems obvious, but the power is lower. In that situation, our chances of being significant are lower, having to do 2 things than 1 thing. It's kind of intuitively makes sense. So I think that's pretty much our time today, and I just want to thank you all for attending. So just in terms of some broad conclusions, like non-inferiority, or NI, and equivalence testing is all about, like, we have a treatment and we want to prove that it's similar to a standard treatment, where with non-inferiority, we want to prove it's no worse than, and with equivalence, we want to prove that it's equal or equivalent to. Non-inferiority, we're usually looking at direct monotonic effects, like where, if it keeps increasing, we're generally going to be happy. And, but we do have the constraint that we usually need to redo something that looks quite similar to the trial that was used to justify the standard due to the assay sensitivity problem. And the NI margin requires careful consideration and obviously discussion with the regulator or any other body that you're dealing with. And, there's kind of a cost-benefit analysis there in terms of, like administration, safety, and stuff like that. 3-arm trials, if they are allowable, due to ethical considerations, allow you to add a direct comparison to placebo, which removes the assay sensitivity problem, and there's a flexible framework available in that case. So just this is a new feature in nQuery added in the last release cycle. So if you're interested in looking at this is available now. And equivalence is all about treating if your treatment is equivalent, but usually, as I say, you can definitely do it on direct effects, and there's no problem with that, but usually looking at indirect effects, such as in bioequivalence studies, where we're looking at AUC, Cmax, these measures of, the blood concentration profile, like how long does it last in the blood, what is the concentration, and ensuring that that's giving similar profile to the original drug. But beware these issues around this the situation with the 90% confidence interval and stuff like that, and some of those issues around and if you're not doing bioequivalence and you're not using these standard regulatory margins, then all of the issues about using science and data to prove what the equivalence limit should be then re-enter the frame as well, of course. So that concludes the webinar. I just want to thank you so much for attending. And if you have any questions you have after this webinar, you can email them to info@statsolves.com, and I'll answer as many of those as possible. And just one final thing I want to mention is that the nQuery summer released a few months ago, including 26 new tables across each area, including multi-arm, multi-stage designs, additional features for phase 2 trials, and including NC-mod, 3-arm trials as mentioned here, cluster randomized swept wedge, confidence interval approaches for proportions, and similar, and survival type tables, including it's an interesting area we're working on survival is in terms of these non-proportional hazards models. And just if you don't have nQuery, or you have a license of nQuery that doesn't include some of the features like adaptive design, you can start a trial at statsolves.com/trial. And we just need if you give an email and sign and get started, you'll be sent and be able to evaluate nQuery within your browser. So you don't need to download anything, you don't need to do anything else, no credit card, anything like that. Just send us just fill in your email here. We'll send you a link, and then you can try nQuery for 14 days within your browser. And it pretty much works as is if it was downloaded on your laptop or computer. And if you want any more additional information, such as our previous webinars, training, or tutorials on nQuery, you can go to statsolves.com/start. And here's just some information, the kind of stuff that we're covering in nQuery these days. As I mentioned, there's references at the end of this demonstration. So if there's anything missing here that you would like more information on, these will obviously be quite useful. So I'm just going to take a couple of moments here to look at any questions that came in today, and then we'll finish up officially. But if you want to leave now, feel free to do so. And if you don't get your question answered in this section, that's fine. I'll be getting back to you via email at a later point, either today or tomorrow, hopefully. Yeah, so there was a couple of questions about basically the non-inferiority problem for other types of data. Like I mentioned the survival case, and as I mentioned, it's not that difficult, because really, when you're doing sample size derivations for survival or proportions or similar, you already have the problem that you need to derive what this test statistics look like under the alternative hypothesis as you move away from, say, a proportion difference of 0 or an odds ratio of 1 or a hazard ratio of 1. So you've already kind of done the work of what the test statistics look like under that. It's just now that you're kind of doing it both ways now. You don't get the nice simple null hypothesis statistic anymore, like usually you're kind of nice centered Z or something like that. But I suppose it's just important to note that whereas it basically, I can actually show this, if we were to take this non-inferiority case for the continuous case and compare that to a 2-sample t-test, obviously we're assuming a t-test for both these cases, and we were to do a one-sided test for superiority, where the actual difference in means was equal to 0.16. So remember, remember, what we're interested in is really this here, the difference between our non-inferiority difference and our expected difference. But if we put in that and we put in our standard deviation of 0.45, the effect size is the same, and we put in the power of 80, you'll get 99 sample size per group again. So for the means case, these are like, this is really just a shifted version of the superiority or inequality type t-test, but where basically the denominator is just equal to, this minus this, or basically this parameter here. But for the proportions case, for the survival case, you can set the standard deviation here independently of that here, but for the proportions case, that's not true. For the survival case, that's not true. For the count case, like negative binomial Poisson, that's not true, which means that the fact that under the null hypothesis, the variance is different than it would be for the nice case where, the difference is equal to 0 or the ratio is equal to 1, that suddenly does come into effect, because oftentimes for those other hypotheses, for those other types of endpoint, the null case, like the ratio equal to 1, often has really nice, simple distributional qualities compared to any other non-null or non-1 or mean difference equal to 0, or sorry, difference equal to 0 situation. But obviously you've lost that now, so you need to account for that. And yeah, and there's just one other question here about equivalence testing. I think they're just mentioning, what was going on here. So basically, this is the concentration of the drug over time. You can see time in the x-axis here. This is on the original scale. This is on the log transform scale. And basically, you can see that obviously, if you administer a drug, the concentration increases, and then it reaches a peak, and then it comes back down. And this is a very classic thing to see. And basically, within this software, you're just selecting the maximum, the C max manually, which is pretty easy to see. It's not hard to see. This is the max one at time 9. And then you can see that it calculates automatically these things like the AUC, and you can see there's like a little distribution here between these like CIs and T, like the concentration, the relationship with concentration and time is given by this linear relationship here. Or sorry, the log concentration is given by this relationship here, and you get all these other distributions like AUCs and T maxes and stuff like that there. So apologies if this is unclear. Like, I know visually it's not great. This is older software, so it's harder to kind of get that involved there. And I think there was just one final question about the HVDs and the NTIDs, kind of asking, these other regulatory situations. So it's far too difficult to probably go in the very brief time I have here, but basically the big difference is that rather than using those standard like equivalence bands, you use equivalence bands that are scaled to the coefficient of variation, certainly for the HVD case, and that also you wouldn't be using a 2x2, you would be using a replicate type design, so like a 2x3 or 2x4, 2x4, and as I say, like the approach you're supposed to use is different for the FDA in the US and for the EMA in the EU. So once again, that adds an extra level of complication. Like, if you're interested in this topic, nQuery covers most of your most of the common situations, like Williams designs and some of these replicate designs. But if you're interested in this topic in general, I do recommend, there is a nice OR package called Power TOST. So Power in lowercase, TOST in capitals, which is probably right now probably the leader in terms of working actively on trying to kind of deal with what seems to be sometimes quite often changing advice on the best way to power for equivalence type designs. Like, really, when you get into the weeds of equivalence testing, it is not trivial in any way. But hopefully you've got a general good overview of what you're mostly generally expected to do, assuming you're not into those complications of NTIDs or highly variable drugs and so on. Okay, I think that's probably enough for now. So once again, for anyone who's still here, thank you so much for attending. And hopefully, if you have any suggestions for future webinars, do let me know in the comments before we finish up. But I think without any further ado, I'd want to thank you so much for attending, and goodbye. So if you have other topics, maybe use this email here. Okay, bye.